And welcome to Group 12's presentation looking at recombinant proteins in medicine. My name is Jo and I'm joined by Alex and Jodlin. During this presentation we will investigate what recombinant proteins are, what the purpose of this technique is and what it's used for, the history and development of the technique, how it actually works, including the current applications of the technique, the advantages and shortcomings, and also future potential applications or extensions of this technique. We will come back to this diagram in a moment, but first let's discuss what recombinant proteins are. A recombinant protein is a type of modified protein whose code is encoded by recombinant DNA. Basically, a recombinant DNA is composed of two segments of DNA joined together in a plasmid, which are generally found in bacteria. When the recombinant DNA is inserted into the bacterial plasmid, they will translate this DNA into proteins which bear the new set of characteristics. The technology of recombinant DNA is possible because of the huge similarity that exists in almost all genetic material of living organisms, and that means that they have similar chemical structure. However, it does not mean that when a recombinant DNA is inserted into a host, it will necessarily express and the protein will be produced. In order to do this, the DNA from the source organism needs to find a vector, and that vector will be the one to drive DNA replication in the host. So what does this mean in simpler terms? Well, a recombinant protein is a synthesized protein that has been cloned in the laboratory from recombinant DNA. Now, this diagram illustrates the central dogma of molecular biology, and you can see from the green arrow that it's possible to produce proteins directly from DNA via test tube. So now let's take a quick look at the history of how this amazing technique came to be. In 1970, a man named Hamilton Smith isolated the first restriction enzyme. This will be covered later in the presentation, but basically a restriction enzyme cuts DNA at a very specific nucleotide sequence. Then, in 1972, Cohen and Boyer create recombinant DNA. In 1976, Boyer then goes on to co-found a firm in the US that is the first to apply recombinant DNA technology. And only 2 years later, in 1978, the first human protein is made using recombinant technology. That protein happened to be somatostatin, which is an important regulator of growth hormone, as you can see from the diagram on the left. However, the first recombinant protein that was made available to the public was actually human insulin. So these are all interesting facts, but the scientific research that went into these discoveries became the greatest race in molecular biology. A fascinating article written by Susan Wright in 1986 looks at the development of this technique from 1972 to 1982. Based on the model of DNA proposed by James Watson and Francis Crick in 1953, by the early 1970s, the genetic code was deciphered, the cellular machinery responsible for replication of DNA and protein synthesis was described in considerable detail, the biochemical pathways involved in replication, expression, and natural recombination were defined, and the enzymes responsible for catalyzing these processes had been isolated. And finally, a model for the regulation of protein synthesis in bacteria was developed and confirmed. These theoretical advances were reflected in new and impressive capacities to manipulate DNA. However, how the DNA in multiple chromosomes of higher organisms, the eukaryotes, functioned, how expression of this DNA was regulated, were understood to be far more complex and difficult problems. The industry was aware of the potential of molecular biology, but were looking for practical applications in medicine, industry, and agriculture. The first step towards an effective method of genetic engineering was DNA splicing, which was achieved in 1971. By obtaining a well-defined piece of DNA and then attaching this piece of DNA to a virus. The next step in development of the technique was the insertion of foreign DNA into bacteria in such a way that it would be replicated. This work was performed by Herbert Boyer and colleagues. At this time, the controlled transfer of foreign DNA into bacteria was brought closer by important observations on the action of the restriction enzyme EcoRI. They showed that the two DNA fragments created by the enzyme would spontaneously join or recombine with one another. In other words, the pieces acted as if their ends were sticky. The significance of this effect was immediately evident. How they could tie DNA from different sources together. However, EcoRI didn't work on a virus, so they started looking for a plasmid, which they later called BCM201. So the next step was immediately clear. The same procedures would be tried with genes from higher organisms. The deliberate, controlled linking of genes from different species was seen as quite a novel and remarkable achievement, and the invention of gene splicing marked the end of biological research as a primarily analytic pursuit and the beginning of synthetic biology. It is at this point that the potential hazards of this technology, both inadvertent and deliberate, started to be contemplated by those molecular biologists conducting the research. However, the research continued with Boyer and colleagues building composite plasmids that would incorporate the most desirable features of several naturally occurring plasmids. For example, the capacity to yield many copies within the cell, the presence of several sites that could be cut by different restriction enzymes, and the capacity to link with very large pieces of DNA. By 1974, researchers still had no idea whether the possible expression of eukaryotic DNA to produce functional proteins when cloned in bacteria was possible. In general, most eukaryotic genes inserted into bacteria before 1977 were not automatically processed and expressed. Whether they could be remained an open question. Then the boom happened. In the period from 1978 to 1982, the number of research projects jumped by 191%, from 546 to 1,588, and financial support itself jumped by 203%, from 61 million to 185 million. Everyone wanted in on this technology, from large multinational corporations, venture capital companies, and fledgling genetic engineering firms. The money to finance this technique poured in, and the race was on to be the first to create a profitable application that was commercially viable. Due to this influx of capital and interest in recombinant DNA technology, those working in the field were able to increase the efficiency and quality of bacterial production of proteins, until finally, after resolving many issues, the appearance in 1982 of the first American product of genetic engineering was the long-anticipated human insulin. So why all this effort to produce recombinant proteins? What is the purpose, and what are they used for? The applications for this technique are many, from medical, agricultural, and industrial applications. Alex will now take you through exactly how this technique works, giving you an example of the production of human insulin, and also give you some specific examples of current applications.
So how does recombinant protein technology actually work? One of the fascinating aspects of DNA is that DNA of one organism, bacteria, can be combined with the DNA of another organism, animal, to make a unique combination known as recombinant DNA or rDNA. Through combining two or more different strands of DNA, scientists can create a new strand of DNA. Usually, this is the combination of human and microorganism. This process is made possible through the use of restriction endonucleases. So overall, recombinant proteins are manipulated forms of protein created in order to produce large amounts of proteins to create a response. These responses have revolutionized medical therapeutic treatments and may be used to create protective antigen-specific immune responses, replace enzymatic deficiencies, and catalytically intervene in disease conditions. So how is this process actually achieved? This process is achieved through a variety of steps. In step 1, rDNA technology first begins with isolating the gene of interest. Next, it is inserted into a vector, where the vectors are then further cloned into multiple copies. But what is a vector? A vector is a section of DNA that is capable of its own independent growth. Bacterial plasmids and viral phages are an example. When the selected DNA of interest is integrated into the vector, this procedure is referred to as rDNA. The next step is step 2. The vector is moved into the host cell. This then leads the host cell to express the coded proteins. The vector must be cloned many times in order to produce adequate amounts of DNA. So the vector is isolated in large quantities so it can be moved into the desired host cells. This then enables the host cells to synthesize the foreign protein in the rDNA. When this process can be produced on a large scale through a bioreactor, this is often known as rDNA technology. Next, we are going to look at this rDNA technology in a little more detail through looking at the steps of the construction of recombinant DNA. In transformation, a select section of DNA is transformed in a vector. Next, that section of DNA is cut with endonucleases, the restriction enzymes, and then ligate the DNA that has been inserted into the vector. This process is known as transformation. Throughout the process of the production of rDNA, E. coli is often the most widely used host organism. Next is non-bacterial transformation. This is a similar process to transformation. In this process, there is no bacteria for the host cell. The process is achieved through a variety of processes, including microinjection, where DNA is inserted into the nucleus of the cell. Another method is known as biolistic transformation, where the host cells are infiltrated with a high velocity of particles coated in DNA. Phage introduction is the process where the difference to the transformation method is that phage is used instead of bacteria. So what are some of the current applications of this technique? Well, there have been many successful applications of rDNA technology since its discovery in the 1970s. rDNA technology has been used for therapeutic proteins in pharmaceutical use, including the use of human proteins such as follicle-stimulating hormone, plasminogen, erythropoietin, growth hormone, and human insulin. The production of biosynthetic human insulin has been a primary breakthrough through the course of rDNA technology history. Recombinant insulin is successfully able to replace insulin in diabetic patients. Following on from the previous slide, a good example of a successful current application of this technique is the use of human insulin. Scientists build the human insulin gene in the lab, where they first begin by isolating the DNA for insulin, and the plasmid made of DNA is removed from the bacterial cell. Restriction enzymes cut the plasmid DNA open, which leaves sticky ends. Next, the insulin gene is added. DNA ligase joins plasmid DNA and insulin DNA. Following this, the vector is then inserted back into the bacterium, and the bacterium cell divides and produces copies of the plasmid. After this process, the vector is now known as recombinant DNA. So this is how the recombinant bacteria uses the gene to produce human insulin. The insulin is then harvested by scientists from the bacteria before being purified and administered to patients.
Hi everyone, my name is Jodeline, and my part is to discuss with you the advantages and shortcomings of the recombinant DNA technology. So when we say recombinant DNA technology, sometimes referred to as genetic engineering, and can benefit people in several ways. For example, scientists made artificial human insulin with the help of recombinant DNA technology. So the advantages of the technique: recombinant DNA in diagnosing infectious disease, help improve health conditions by newly developed vaccines and pharmaceuticals, and it's a treatment of diseases. We're going to discuss first the recombinant DNA in diagnosing infectious disease, using cloned genes for E. coli, heat labile, and thyrotoxin. So Maselli and co-workers have shown that DNA hybridization is a powerful tool to detect the presence of a pathogen directly in clinical specimens. So another one is the DNA probes, have been constructed that detect chlamydia trachomatis and gonorrhea and a variety of enteric pathogens. So speaking of chlamydia trachomatis, is an obligate intracellular bacterium whose only natural host is humans. So although presenting as asymptomatic in most women, genital tract chlamydial infections are a leading cause of pelvic inflammatory disease, tubal factor infertility, and ectopic pregnancy. And another one is the DNA hybridization. So it has been used to detect cytomegalovirus DNA in urine. Cytomegalovirus is a member of the herpes family. Related viruses include Epstein-Barr, causes glandular fever; varicella zoster, causes chickenpox; and herpes simplex, like causes cold sores. So this viral infection can be spread through coughing, contact with blood, urine, or feces, or via the mucous membranes, such as the mouth and genitals. DNA probes could also be used as a rapid diagnostic adjunct to tissue culture or conventional bacterial culture techniques. Another advantage of DNA technology is to help improve health conditions by newly developed vaccines and pharmaceuticals. The classical example of recombinant protein vaccines currently used in humans is the vaccine against hepatitis B. Hepatitis B virus infection is a chronic liver disease occurring worldwide. So hepatitis B vaccine is a vaccine production in plants. So plants have been used to produce several therapeutic proteins products, such as casein and lysozymes for improving health of child and polymers of protein for tissue replacement and surgery. Hepatitis B vaccination programs using either Angerix B or Recombivax HBvax2 can achieve high seroprotective response rates, particularly in childhood and adolescence. So let's continue discussing the advantages of DNA technology. So DNA technology can treat different diseases. Like for example, the Lespro, in comparison with regular human insulin, is a well-effective and fast-acting recombinant insulin. The FFN alpha is a novel and will recognize recombinant protein as well that can be effectively used in curing of anemia. And recombinant human growth hormone was found with a great improvement in treating children lacking the ability to produce human growth in a required quantity. So the production of recombinant human growth hormones by recombinant DNA technology produced by the pituitary gland. So human growth hormone is essential for proper growth in children. Let's discuss now the shortcomings of recombinant DNA technology. So we have the functional deficiency in proteins, gene therapy, and the gene doping. So the first shortcoming is the functional deficiency in proteins. Common obstacles which must be dealt with are post-translational modifications, cell stress responses activation, and instability of proteolytic activities, loose solubility, and resistance in expressing new genes. And mutation occurring in humans at genetic level cause deficiency in proteins as well, so which can be altered or be treated by incorporation of external genes to fill the gaps and reach the normal levels. Another shortcoming is the gene therapy. In gene therapy, a normal gene is inserted into the genome to replace an abnormal gene responsible for causing a certain disease. So the various challenges involved in the process, one of the most significant is the difficulty in releasing the gene into the stem cell. Thus, a molecular carrier called a vector is used to release the gene, which needs to be very specific, display efficiency in the release of one or more genes of the sizes necessary for clinical applications, not be recognized by the immune system. So, although the efficacy of viral vectors is confirmed, recently some studies demonstrated that the use of these carriers represented with several limitations. In terms of gene doping, according to the list of prohibited substances published by WADA in 2008, gene doping has been defined as non-therapeutic use of cells, genes, genetic elements, or modulation of gene expression having the capacity to enhance athletic performance. In 2013, WADA clarified the type of manipulation of genetic material prohibited in sport as the transfer of nucleic acid or their analogs into cells and the use of genetically modified cells. So gene doping can be performed without the use of security and protective measures. So vectors for gene transfer produced in uncontrolled laboratory conditions can be contaminated. Example by chemical and or biological agents, so thereby endangering the health and life of athletes. Before we end our presentation, I would like to talk to you about potential future applications or extensions of the technique. So future protein products might include cancer vaccines and therapies for a variety of genetic diseases, live recombinant vaccines using bacterial or viral vectors, and recombinant DNA vaccine technology for animal and human vaccination. So there were three different types of preparation based on recombinant DNA vaccine technology that are used for animal and human vaccination. So first is genetically modified viruses. Scientists have used RNA vaccine technology to genetically modify live viruses so that they can still elicit an immune response, but not be pathogenic. So this requires knowing what genes in the virus are associated with viral replication and subsequently deleting or knocking those genes out. And in terms of recombinant viral proteins, for those viruses in which the protein or antigen that induces the immune response is known, the viral DNA that codes for that particular protein can be isolated, cloned, and used to make viral protein in a test tubes. And genetic vaccines are comprised of stripped down pieces of viral DNA that are engineered to initiate the expression of a protein antigen specific to the disease after injection into the animal undergoing vaccination. So these small viral DNA pieces are injected under the skin, after which the host cells take up the DNA. The DNA template is translated and viral proteins are made within the host cells. The host immune system reacts as if it's been exposed to the disease itself and tries to fight it by making antibodies against the newly synthesized viral proteins. So, once again, thank you for watching and hope you enjoyed our presentation.